Let us continue our video lecture on transport of ions and macromolecules. In this video, we will study about the intracellular vesicular traffic across endoplasmic reticulum and Golgi bodies. Now, every cell must eat, communicate with the world around it, and quickly respond to changes in its environment. To help accomplish these tasks, cells continually adjust the composition of their plasma membrane in rapid response to need. They use an elaborate internal membrane system to add and remove cell surface proteins embedded in the membrane, such as receptors, ion channels, and transporters. Though the process of exocytosis, the biosynthetic secretory pathway delivers newly synthesized proteins, carbohydrates, and lipids to either the plasma membrane or the extracellular space. By the converse process of endocytosis, cells remove plasma membrane components and deliver them to internal compartments such as endosomes, from where they can be recycled to the same or different regions of plasma membrane. They can also be delivered to lysosomes for degradation. Cells also use endocytosis to capture important nutrients such as vitamins, lipids, cholesterol, and iron. These are taken up by together with the macromolecules to which they bind and are then released in the endosomes or lysosomes and transported into the cytosol, where they are used in various biosynthetic processes. The interior space or lumen of each membrane enclosed compartment along the biosynthetic secretory and endocytic pathway is topologically equivalent to the lumen of most other membrane enclosed compartments and to the cell exterior. Proteins can travel in this space without having to cross a membrane, being passed from one compartment to another by means of numerous membrane enclosed transport containers. Some of these containers are small spherical vesicles, while others are large irregular vesicles or tubules formed from the donor compartment. We will use the term here transport vesicle to apply to all forms of these containers. Within a eukaryotic cell, transport vesicles continually bud off from one membrane and fuse with another, carrying membrane components and soluble molecules which are referred to as cargo. This membrane traffic flows along highly organized directional routes which allow the cell to secrete, eat, and remodel its plasma membrane. The biosynthetic secretory pathway leads outward from the endoplasmic reticulum towards Golgi apparatus and cell surface, with a side route leading to lysosomes, while the endocytic pathway leads inward from the plasma membrane in each case. The flow of membrane between compartments is balanced, with retrieval pathway balancing the flow in the opposite direction, bringing membrane and selected proteins back to compartment of origin. To perform its function, each transport vesicle that buds from a compartment must be selective. It must take up only the appropriate molecules and must fuse only with the appropriate target membrane. A vesicle carrying cargo from the Golgi apparatus to the plasma membrane, for example, must include proteins that are to stay in Golgi apparatus. It must fuse only with the plasma membrane and not with any other organelle. We begin this video by discussion on the molecular mechanism of budding and fusion of underlying all vesicular transport. We will then move on to the fundamental problem of how, in the face of this transport, the cell maintains the differences between compartments. Finally, in the end part of the videos, we will learn the function of Golgi apparatus, lysosomes, secretory vesicles, and endosomes. Let us firstly understand the molecular mechanism of membrane transport and the maintenance of compartmental diversity. Vesicular transport mediates a continuous exchange of components between the 10 or more chemically distinct membrane enclosed compartments that collectively comprise the biosynthetic secretory and endocytic pathways. In this massive exchange, how each how can each compartment maintain its specific identity? To answer this question, we must consider what defines the character of a compartment. Above all, it is the composition of the enclosing membrane. Molecular markers displaced on the cytosolic surface of the membrane serve as guidance, cues for incoming traffic to ensure that the transport vesicles fuse only with the correct compartment. Many of these membrane markers, however, are found on more than one compartment, and it is the specific combination of marker molecules that gives each compartment its unique molecular address. How are these membrane markers kept at high concentration on one compartment and at low concentration on another? To answer this question, we need to consider how patches of membrane enriched or depleted in specific membrane components bud off from one compartment and transfer to another. We begin the discussion by how cells segregate proteins into separate membrane domains by assembling a special protein coat on membrane cytosolic phase. We will consider how coats form, what they are made up of, and how they are used to extract specific components from a membrane for delivery to another compartment. Finally, we will discuss also about transport vesicles dock at the appropriate target membrane and fuse with it to deliver their cargo. Let us understand about firstly various types of coated vesicles. Yes. Now, most transport vesicles form from specialized coated regions of membranes. They bud off as coated vesicles which have a distinct cargo of proteins covering their cytosolic surface. Before the vesicles fuse with the target membrane, they discard their coat as is required for the two cytosolic membrane surface to interact directly and fuse. The coat performs two main functions. First, it concentrates specific membrane proteins in a specialized patch, which then give rise to vesicle membrane. In this way, it selects the appropriate molecules for transport. Second, the coat molds the forming vesicle. Coat proteins assemble into curved basket-like lattice that deforms the membrane patch and thereby shape the vesicle. This may explain why vesicles with the same type of coat often have relatively uniform size and shape. There are three well characterized types of coated vesicles, distinguished by their coat proteins: Clathrin coated, Cobb coated, and Cobb2 coated. Each type is used for different transport steps. Clathrin coated vesicles, for example, mediate transport from Golgi apparatus and from the plasma membrane, whereas COP1 and COP2 coated vesicles most commonly mediate transport from ER and from the Golgi cisternae. There is, however, much more variety in coated vesicles and their functions than this short list suggests. We will also discuss about the several types of Clathrin coated vesicles, each specialized for different transport step, and the COP1 and COP2 coated vesicles may be similarly diverse. Clathrin coated vesicles, the first coated vesicle to be identified, transport material from plasma membrane and between endosomal and Golgi compartments. The COP1 coated vesicle and COP2 coated vesicle transport material early in the secretory pathway. COP2 coated vesicle bud from ER and COP1 coated vesicle bud from Golgi compartments. The major protein components of clathrin coated vesicle is clathrin itself. Each clathrin subunit consists of three large and three small polypeptide chains that together form three-legged structure called as triskelion. So what is the name? What is it is called? Triskelion. Clathrin triskelion assemble into a basket-like convex framework of hexagons and pentagons to form coated pits on the cytosolic surface of membrane. Under appropriate conditions, isolated triskelions spontaneously self-assemble into typical polyhedral cages in a test tube. Even in the absence of the membrane vesicle that these baskets normally enclose. Thus, the clathrin coated triskelions determine the geometry of the clathrin cage. Adapter proteins, another major coat component in the clathrin coated vesicle, form a discrete second layer of coat positioned between the clathrin cage and the membrane. They bind the clathrin coat to the membrane and trap various transmembrane proteins including transmembrane receptors that capture soluble cargo molecules inside the vesicle, so-called cargo receptors. In this way, a selected set of transmembrane protein together with the soluble proteins that interact with them are packaged into each newly formed clathrin coated transport vesicle. There are several types of adapter proteins. The best characterized have four different protein subunits. Others are single chain proteins. Each type of adapter protein is specific for different set of cargo receptors and its use leads to formation of distinct clathrin coated vesicle. Clathrin coated vesicle budding from different membranes use different adapter proteins and thus package different receptors and cargo molecules. The sequential assembly of adapter molecules and a clathrin coat on the cytosolic surface of membrane generate forces that result in the formation of a clathrin coated vesicle. Lateral interaction between adapter molecules and complexes and between clathrin molecules aid in forming the vesicle. Now, not all coats that we form are regular and universal as example of clathrin or COP coat suggest. Some coats may better be described as specialized protein assemblies that form patches dedicated to specific cargo proteins. An example is a coat called retromer which assembles on endosomes and forms vesicles that return acid hydrolase receptors such as mannose-6-phosphate receptor to the Golgi apparatus. Retromer is a multi-protein complex that assembles into a coat on endosomal membrane only when it can bind to the cytoplasmic tails of cargo receptors. It can interact directly with a curved phospholipid bilayer and it can bind to a specific phosphorylated phosphatidyl inositol lipid which acts as endosomal marker as we will discuss in the coming slides. Because these three requirements must be met simultaneously, a retromer is thought to act as a coincidence detector and only assemble at a right time and right place. Upon binding as a dimer, it stabilizes the membrane curvature which makes binding of additional retromers in its proximity more likely. The cooperate assembly of retromer then leads to the formation of formation and budding of transport vesicles which deliver its cargo to the Golgi apparatus. The adapter proteins found in the clathrin coats also bind to phosphoinositides, which not only have a major role in directing when and where coat assemble in the cell, but are used much more widely as molecular markers of compartment's identity. This helps to control membrane trafficking events. Let us firstly now understand how phosphoinositides mark organelle and membrane domains. Although inositol phospholipids typically comprise less than 10% of total phospholipids in membrane, they have important regulatory functions. They can undergo rapid cycles of phosphorylation and dephosphorylation at 3' and 5' positions of their inositol sugar head groups to reproduce various types of phosphoinositides. It is short form, it is called as PIPs. The interconversion of phosphatidyl inositol and PIPs is highly compartmentalized. Different organelles in the endocytic and biosynthetic secretory pathways have distinct set of PIs and PIP kinases and PIP phosphatases. The distribution, regulation and local balance of these enzymes determine the steady state distribution of each PIP species. The distribution of PIP varies from organelle to organelle and often within a continuous membrane from one region to another, thereby defining specialized membrane domains. Many proteins involved at different steps in vesicular transport contain domains that bind with highly specificity to the head group of particular PIPs, distinguishing one phosphorylated form to another. Local control of the PI and PIP kinases and PIP phosphatases can therefore be used to rapidly control the binding of proteins to a membrane or membrane domain. The production of a particular type of PIP recruits proteins containing matching PIP binding domains. The PIP binding proteins can help regulate vesicle formation and another step in membrane transport. The same strategy is widely used to recruit specific intracellular signaling proteins to the plasma membrane in response to extracellular signals. Now, let us go back to Golgi apparatus and the endoplasmic reticulum. See, when we are talking about transport from the endoplasmic reticulum through the Golgi apparatus, the newly synthesized proteins that cross the plasmic reticulum membrane from the cytosol to enter the biosynthetic secretory pathway. During their subsequent transport from ER to Golgi apparatus and from Golgi apparatus to cell surface and elsewhere, these proteins are successfully modified as they pass through series of compartments. Transfer from one compartment to the next involves a delicate balance between forward and backward retrieval transport pathways. Some transport vesicles select cargo molecules and move them to next compartment in the pathway, while others retrieve escape proteins and return them to previous compartment where they formerly function. Thus, the pathway from the ER to the cell surface consists of many sorting steps which continuously select membrane and soluble luminal proteins from packaging and transport in vesicles or organelle fragments that bud from ER and Golgi apparatus. In this video slide, we would mainly focus on Golgi apparatus, also called as Golgi complex. Now we know that Golgi apparatus or Golgi site is a major area of carbohydrate synthesis as well as sorting and dispatching station for products of the ER. The cell makes many polysaccharides in the Golgi apparatus, including pectin, hemicellulose of the cell wall in the plants, and most of the glycosamine glycans of the extracellular matrix in the animals. The Golgi apparatus also lie on the exit route from the ER, and a large proportion of the carbohydrates that it makes are attaches as oligosaccharide side chains to the many proteins and lipids that the ER sends to it. A subset of these oligosaccharide groups serve as tags to direct specific proteins into vesicles that then transport them to lysosomes. But most proteins and lipids, once they have acquired their appropriate oligosaccharides in the Golgi apparatus, are recognized in other ways for targeting into transport vesicles going to other destinations. Now, if we talk about how proteins leave ER, now proteins leave ER in COP2 coated transport vesicles. We have already discussed about the calcium coated COP1 and COP2 coated vesicles. To initiate their journey along the biosynthetic secretory pathways, proteins that have entered the ER and are destined for the Golgi apparatus or beyond are first packaged into small COP2 coated transport vesicles. These vesicles bud from specialized regions of the ER called ER exit sites, whose membrane lack bound ribosomes. Whose membrane lacks bound ribosome. Most animal cells have ER exit sites dispersed throughout the ER network. Originally, it was thought that all proteins that are not tethered in the ER enter transport vesicles by default. It is now clear, however, that the entry into vesicles that leave the ER is usually selective process. Many membrane proteins are actively recruited into such vesicles where they become concentrated. It is thought that these cargo proteins display exit transport signals on their cytosolic surface that components of COP2 recognize. These code components act as cargo receptors and are recycled back to ER after they have delivered their cargo to Golgi apparatus. Soluble cargo proteins in the ER lumen, by contrast, have exit signals that attach them to transmembrane cargo receptors, which in turn binds through exit signal in their cytoplasmic tails to components of the COP2 coat. At a lower rate, without proteins exit signals and can also enter transport vesicles, so that even proteins that normally function in the ER or so-called ER resident proteins slowly leak out the ER and are delivered to the Golgi apparatus. Similarly, secretory proteins that are made in high concentration may leave the ER without the help of exit signals or cargo receptors. The exit signals that direct soluble proteins out of the ER for transport to the Golgi apparatus and beyond are not well understood. Some transmembrane proteins that serve as cargo receptors for packaging some secretory proteins into COP2 coated vesicles are lectins that bind to oligosaccharides. The ERG1C53 lectin, for example, binds to mannose and is thought to recognize this sugar on the two secreted blood clotting factors, factor 5 and factor 8, thereby packaging the proteins into transport vesicles in the ER. ERGIC53's role in protein transport was identified because humans who lack it, owing to an inherited mutation, have lowered serum levels of factor 5 and 8 and they therefore bled excessively. Now, what are what are actually the proteins that has to be the proteins that leave that has to be properly folded and assembled, and only then they can leave the ER. To exit from the ER, proteins must be properly folded, and if they are subunits of multimeric protein complexes, they may need to be completely assembled. Those are misfolded or incompletely assembled remain in the ER, but they bound to chaperone proteins such as BIP or calnexin. The chaperones may cover up the exit signals or somehow anchor the proteins in the ER. Such failed proteins are eventually transported back into the cytosol where they are degraded by proteasomes. This quality control step prevents the outward transport of misfolded or misassembled proteins that could potentially interfere with the function of normal proteins. There is a surpassing amount of corrective action. More than 90% of the newly synthesized subunits of T cell receptor and of the acetylcholine receptor for example, are normally disregarded without ever reaching the cell surface where they function. The cells must make a large excess of many protein molecules to produce select few that fold, assemble, and function properly. The process of continual degradation of a portion of ER proteins also provides an early warning system to alert the immune system when a virus infects the cells. Using specialized ABC type transporters, the ER imports peptide fragments of viral proteins produced by proteases in the proteasome. The foreign peptides are loaded onto class 1 MHC proteins in the ER lumen and then transported to the cell surface. T lymphocytes then recognize the peptides as non self antigens and kill the infected cells. Sometimes, however, there are drawbacks to the stringent quality control mechanism. The predominant mutations that cause cystic fibrosis, a common inherited disease, result in the production of slightly misfolded form of plasma membrane protein important for chloride transport. Although the mutant protein would function perfectly normally if it reaches the plasma membrane, it remains in the ER. This devastating disease thus results not because the mutation inactivates the protein, but because active protein is discarded because it reaches the plasma membrane. Now, after the transport vesicles, so you can see in the video, here it is the budding of the transport vesicles. So after the transport vesicles have budded from ER exit sites and have shed their coat, they begin to fuse with one another. This fusion of membranes from same compartment is called as homotypic fusion to distinguish it from heterotypic fusion, in which the membrane from one compartment fuses with the membrane of a different compartment. As with the heterotypic fusion, homotypic fusion requires a set of matching snare. In this case, however, the interaction is symmetrical with both membranes contributing V snare and T snares. The structures formed when ER derived vesicles fuse with one another are called as vesicular tubular clusters because they have convoluted appearance in the electron microscope. These clusters constitute a new compartment that is separated from ER and lacks many of the proteins that function in the ER. They are generated continuously and function as transport containers that bring material from ER to the Golgi apparatus. The clusters are relatively short lived because they move quickly along microtubules to the Golgi apparatus with which they fuse to deliver their contents. As soon as vesicular tubular clusters form, They begin to bud of transport vesicles of their own. Unlike the COP2 coated vesicles that bud from ER, these vesicles are COP1 coated. They carry back to the ER resident proteins that have escaped, as well as proteins such as cargo receptors that participated in the ER budding reaction and are being returned. This retrieval process demonstrates the exquisite control mechanism that regulate coat assembly reactions. The COP1 coat assembly begins only seconds after COP2 coats have been shed. It remains a mystery how this switch in coat assembly is controlled. The retrieval transport continues as the vesicular tubular clusters move towards the Golgi apparatus. Thus, the clusters continuously mature, gradually changing their composition, and selected proteins are returned to the ER. See, the retrieval pathway to the ER uses sorting signals. So, suppose some protein that has to be retrieved back to the ER because it is the ER resident protein that can be present in either the ER membrane or the cytoplasm of the ER. They use a certain sorting signal so that to prevent them from excreted out of the Golgi apparatus. The retrieval pathway for returning escaped proteins back to the ER depends on ER retrieval signals. Resident ER proteins, for example, contain signals that bind directly to COP1 coated coats and are thus packed into COP1 coated transport vesicles for retrograde delivery to the ER. The best characterized retrieval signal on this type consists of two lysines followed by two other amino acids at the extreme C terminal end of the ER membrane protein. It is called as KKXX sequence based on single letter amino acid code. So we know KK is lysine and XX can be any other amino acid. Soluble ER resident proteins such as BIP also contains short retrieval signal at the C terminal end, but it is different. It consists of a lysine, aspartate, glutamate, leucine or similar sequence. If this signal different from the KDL sequence is removed from BIP by genetic engineering, the protein is selectively or slowly secreted out of the cell. If the signal is transferred to a protein it is normally secreted, the protein is now efficiently returned back to ER when it accumulates. Unlike the iterative signals on ER membrane proteins, which cannot interact directly with the COP1 coat, soluble ER resident proteins must bind to specialized receptor present such as KDEL receptor, a multipass transmembrane protein that binds to KDEL sequence and packages any protein displaying it into COP1 coated retrograde transport vesicles. To accomplish this task, the KDA receptor itself must cycle the ER and Golgi. The receptor must have high affinity for KDL sequence in vesicular tubular clusters in the Golgi apparatus so as to capture escaped soluble ER resident proteins that are present there are at low concentration. It must have low affinity for the KDL sequence in the ER, however, to unload its cargo in spite of very high concentration of KDL containing resident proteins in the ER. How does the affinity of KDL receptor change depending on the compartment in the which it resides? The answer is not much known, but it may be related to the different iconic conditions and pH, different ionic condition, pardon, and pH in different compartments, which are regulated by ion transporters in the compartment membrane. As we discuss later, pH sensitive protein protein interaction form the basis for many sorting steps in cell. Most membrane proteins that function in the interface between ER and the Golgi apparatus include V and T snares. Whereas the recycling of some of the proteins is mediated by signals, for others no specific signal seems to be required. Thus, while retrieval signal increase the efficacy of the retrieval process, some protein randomly enter budding vesicle destined for the ER and return to the ER at a slower rate. Many Golgi enzymes cycle constantly between ER and Golgi, but their rate of return to ER is slow enough for the most of the protein to be found in Golgi apparatus. Many proteins are selectively retained in the compartments in which they are function. The KDL retrieval pathway only partly explains how ER resident proteins are maintained in the ER. As expected, cells that express genetically modified ER resident proteins from which the KDL sequence has been experimentally removed secrete these proteins. We know that Golgi has cis phase and trans phase. The Golgi apparatus transport molecules undergo ordered series of covalent modification. Each Golgi stack has two phases, cis phase or trans phase. Cis phase is entry phase and trans phase is exit phase. Now, protein transport can be from RER to Golgi apparatus. They are packed to secretory vesicle and target organelle or exterior of the cell. Now, proteins to be destined to ER, Golgi apparatus, lysosome, and plasma membrane, and cell exterior are translated on bound ribosomes. Proteins to be destined to cytosol, mitochondria, chloroplast nucleus are translated on free ribosomes. So this is the diagram of a co-translational modification or a co-translocation of protein, while the other is post-translational modification. Post-translational modification means that the proteins are transported after they have been completely translated, while the co-translation means that while synthesizing of the proteins, it is simultaneously entered into the rough endoplasmic reticulum. Now, the proteins bind to signal receptor particles which help in translocation of protein inside the ER. Now, after once the protein has been packed, it enters the Golgi apparatus where it is glycosylated. Glycosylation is of two types, N-linked and O-linked. When the sugar residues are added to the asparagine residue of the proteins, it becomes N-linked glycosylation. While if it is added to serine threonine residues, it is called as O-linked glycosylation. Now, carbohydrate residues to be transferred are held by a special molecule called as dolichol phosphate, and then these carbohydrates residues are transferred to asparagine residue of protein by membrane-bound oligosaccharide transferase enzyme. Now, proteins to be exported are categorized in two ways. Protein that to be retrieved back, as we have studied, example is BIP, but the protein that is further packed and targeted to destined granule organelle, and protein to be retrieved can be either ER resident membrane protein with KKXX sequence or ER resident soluble protein with KDL sequence. So this was the type of two vesicles that we have already studied. Once the protein has to be translocated to a particular target tissue, smear membrane proteins are present on the vesicles.
There are two types of snare, V snare and T snare, as we have already studied. V snare is present on the vesicle and T snare is present on the target membrane, and both has to be fused together to form two membrane fuses, like together in two membrane fuses, and protein cargo is translocated to target organelle. So this was all about the translocation of proteins across the ER and the Golgi apparatus.